Our first speaker will be discussing wheat breeding for the Canadian Prairies. And Santosh Kumar, the wheat breeder from Agriculture and Agri-Food Canada. Dr. Santosh Kumar is a spring wheat breeder with Agriculture and Agri-Food Canada, as I mentioned, and he's stationed in Brandon here at the Research and Development Centre. Dr. Kumar has published in areas of plant physiology, molecular biology, genetics, and genomics. The current focus is on developing premium quality wheat cultivars to improve the profitability for farmers, marketers, and processors. The BRDC breeding program and the Cereal Genomics Laboratory combines traditional breeding methods with new marker technologies to improve breeding efficiencies in a cost-effective manner. Since joining AFC, BRDC 5 years ago, Dr. Kumar has participated in the registration of 7 varieties. Dr. Kumar is actively involved in all aspects of wheat research in Canada. Please welcome Dr. Kumar.
Good morning, everyone, and thank you for coming to this talk. It's really great to be a wheat breeder in the wheat city. I'll start with that. I'm a research scientist with Agriculture and Agri-Food Canada, and thank you for the kind introduction. By that, I meant I have tried many different things. I'm not really good at anything, but I do a lot of different things. So that's what breeding is, actually. So briefly, I'll be talking about the production statistics of wheat in Canada, especially in the Western Canadian perspective. I'll talk a little bit about the wheat breeding research that we do at the BRDC, and a little bit of a glimpse about what we do at the AFC overall at different centers. What are the technologies that we use in breeding? So breeding is not all about just going to the field and watching the plants grow. It's a lot about different technologies that improve the efficiency of breeding. So a little bit of information on that. And then what are the new innovative technologies that accelerate the rate of gains of breeding for any given breeder? So a little bit on that. Okay. So this is the production statistics as of 2019. As you can see, wheat is a major crop across the Western Canadian provinces. Within the cereals, wheat has the maximum acreage of about 14.5 million acres, followed by barley, then durum, oats, and corn. So there is a significant gap in the cereal acreage from wheat to any other cereals. So it is safe to say that wheat commands a lot more acreage in the Western Canadian provinces. This is the wheat acreage in the 3 prairie provinces, with Saskatchewan leading within the provinces at about 8.44 million acres, followed by Alberta, and then Manitoba. British Columbia contributes very little to the wheat acreage. These are different classes of wheat that are grown in Canada, especially in the Western Canadian perspective. CWRS is the premium quality wheat, the Canada Western Red Spring, followed by Canada Western Amber Durum, Canada Prairie Spring, the soft whites. This is a new class of wheat that came into being about 3 years ago when we had challenges with gluten strength because of a lot of the northern United States lines coming to Canada, and we wanted to find a place for them. So this class of Canada Northern Hard Red was created to account for lines that do not have sufficient gluten strength and extensibility. All right, moving on. So this is The production of wheat in 2019, that was about 32 million metric tons, and we exported about 15 million metric tons, contributing about $6 billion to the Canadian GDP. That's a significant amount for any country to export from the premium quality wheat. And Canada leads the premium quality wheat market. Look how the acreages have changed over the years, over the last 2 years. Durum acreage has decreased significantly by 21%, and that's because the harsh growing conditions plus the fusarium pressure that is moving westwards from Manitoba. The spring wheat acreage has gained by 14%. Needless to say, it's a more resilient type of wheat. It's a hexaploid, so the genome has a lot more plasticity for acclimatizing to the harsh conditions. And that's why it's an increased acreage. Winter wheat has decreased significantly because of the winter kill. But overall, we see just a minor decrease in the yield, which is compensated by the spring wheat for the loss in acreage because of durum and winter. These are wheat exports from Canada, or the world exporters. These countries contribute about 75% of the total wheat exports all over the world. And Canada is just above United States at about 13.8% of the total wheat exports. Most of these exports are coming out of the CWRS class wheat, which is the premium quality wheat. So I'm extremely proud to say that I breed the Western hard red spring. Now talking about the research component of wheat breeding. This is a diagram that shows the different steps of breeding. So we start with 2 parents. The choice of parent depends on what traits they have. Usually, a breeder tries to bring in 2 different parents that are having complementary traits. So if one is lacking fusarium resistance and another one is lacking leaf rust resistance, we bring them together to complete that package of good disease resistance, both for fusarium and leaf rust, and then bring in together to make the F1s. These F1s go through 2 different pathways. One can go through a traditional selfing, which the plant is allowed to make seed and move on to the next generation. That's called selfing. Or we can send it to double haploids. In double haploids, the F1s are actually, the chromosomal complement is reduced to half so that we don't get any segregation of the traits going forward in the later generations. And the chromosome is just doubled by application of a chemical called colchicine. It allows us to breed better varieties faster. That's what it is. Once we have done that, either through the double haploid pathway or the regular selfing pathway, we do early generation testing, which means we grow yield plots, we do disease testing, we do quality testing. And within those testings, for ergonomic traits, we look at yield, protein, maturity, height, and lodging. For diseases, we look at the 5 primary diseases that we have to have minimum level of intermediate resistance. They are the 3 rust, the leaf rust, stem rust, and stripe rust, fusarium head blight, and common bunt. We also look at quality objectives. There are 16 or 17 different traits that we look at, but they are classified under the milling traits, the dough properties, and the baking attributes. Once the early generation testing is done, we'll move the lines into advanced testing where we grow them at multiple locations to look at the plasticity of yield at multiple locations under different soil conditions, climatic conditions, disease pressure. That's what advanced generations are. And in every generation, we make a lot of culls. So in general, we throw away about 80 to 90% of the germplasm that is grown in a given year. And we move forward 10% of the germplasm. So for example, if we start with 250,000 plants at F2s, by F6s, we are left at about 600. So that's the rate of cull. So we go through a lot of plants in looking at them individually and making selections for different traits that are outlined here. Once the advanced generation testing is done, these lines get put into registration trial, which is called as the variety registration trial, and it is mandated by the Prairie Grain Development Committee, which works under the CFIA guidelines, to do this registration trials. So the lines that are promising are put through these registration trials for 3 years, and it's done in an impartial way, and all the data for all these traits here again are collected and compiled in a report at the end of the registration trial after 3 years. This report is submitted to the Prairie Grain Development Committee that does the support of registration after rigorous assessment of the data. If a line is supported, that's the real work, then the real work begins on that specific lines, because you have to do the plant breeder rights documentation, the variety registration. Meanwhile, all of this is going on, the varieties that is licensed out to different needs, they start growing it from the breeder seed all the way up to certified seed, and that takes about 3 to 5 years. That is your breeding pipeline, and after that process of certified seed production, that's when you see it in the farmer's field. About 12 to 15 years. So yes, we make sure that the line is really good before it gets to you. Looking at different objectives, first one is agronomic objectives. There is a superior yield requirement. So when I joined the programs 5 years ago, I asked a farmer, what is your wish list for me to do? And he said, well, you should look at yield, and you should look at yield, and then you should look at yield. And I got the answer there that yield is our primary target. If the yield doesn't perform, the line doesn't perform in terms of yield, the farmers don't make money. Then he went on saying that you should have very good quality, okay? And the disease package should be such that I don't need to spray. I said, I got my answer. So we work on all of them. So we need superior yield for sure. We have to maintain the maturity. That's a critical one, because if we allow the maturity to drag, you are into snow, you are into bad weather, and that degrades the quality of your grain. A lot of breeding effort has gone into maintaining that maturity, because as you allow the maturity to drag on, you do get a yield advantage, you get the protein, but you can't harvest the crop sometimes because of the bad weather. So the maturity has to be at that 99 days, 98 days, and it can't be dragged on. So we have to make that yield and protein gains while maintaining that maturity, days to maturity. We need the lodging resistance, because as we go on to the high-yielding crops, the spike and the head of the crop gets heavy with heavy grain. And if there is bad weather, high winds, a lot of moisture, it tends to lean down. If it leans down and it's top heavy, it doesn't come up. So the lodging becomes a major challenge during harvest. Shorter straw. Farmers don't want to deal with a lot of straw. So we are trying to develop more and more varieties that have stronger straw and short straw. So you don't have a lot of straw management to do at the end of the harvest. We need the higher test weight to compete in the market. Pre-harvest sprouting resistance, as the discussion started already, it's a key component of our program. We invest a lot of time in making sure that the lines have pre-harvest sprouting resistance. And that's what allows the great protection of the crop, because if there is pre-harvest sprouting, the lines do not germinate and start degrading that amylase at the end of the life cycle. That's what protects the grade of the crop. So we invest a lot on the pre-harvest sprouting resistance of the crop. The major diseases that we work on are rusts. So there is the leaf rust caused by Puccinia tritici. The stripe rust is caused by Puccinia striformis. Stem rust by Puccinia graminis. And the Fusarium head blight, which is the major problem for us, is caused by Fusarium graminearum. So these are the major diseases that we focus on. We also focus on common bunt. And these 5 diseases are the priority diseases for us. But we do not discount the loose smut and leaf spot, because the genetic resistance to these diseases is the best resistance, because if you're not spraying for any of these diseases, why would you spray for loose mud? So we try to incorporate the loose mud and leaf spot disease resistance as well, even though it's not a requirement for registration of a variety. For quality assessment, we look at the protein content, which is of key prime importance, because this protein content not only determines the quality or the grade of the crop and the classification of the crop, it also affects the gluten strength, the gluten extensibility, and water absorption of the crop. So the protein content is important in many different aspects of the quality of wheat grain. We look at loaf volume after baking. We don't directly work on the food quality, but we work with different food scientists to look at traits such as asparagine content. Asparagine is an amino acid, and if you have a lot of asparagine into the wheat grains, then it leads to formation of acrylamide, which is not healthy for human consumption. So we work with different food scientists to screen our germplasm so that we screen only the lines that are having lower concentration of asparagine amino acid in the crop. So we actively work on that as well. Okay. So coming on to what resources do we use? What is the resources that we have for doing breeding? So we are located at Brandon, and this program looks at the premium quality wheat and the oat breeding. Our satellite location is at Saskatoon. We work at Beaver Lodge. So this location at Brandon looks after two different types of wheat classes. One is the CWRS for the Eastern Prairies, which is east of Saskatoon. Another one is the Parkland program, which is north of Saskatoon, and both programs are operated out of Brandon. There is another wheat breeding program at Swift Current that does a lot of the wheat breeding for the Western Prairies, the western part of the Prairies. We have stations at Lethbridge that does stripe rust testing and bun testing for us. We actively work with researchers in Ottawa and Charlottetown, especially for fusarium resistance. Because if you know the history of how fusarium actually became a challenge for us, the 3A Dawn type fusarium graminearum came out of the eastern part of Canada and became a major challenge for us. As it moves westwards, it is going to be a major challenge in Saskatchewan and Alberta as well. But we actively look at these sites so that we can account for the new races of fusarium that is coming to Canada. That's why the nurseries at Ottawa and Charlottetown become so much more important for us. The Brandon site operates at about 7 sites that we maintain and manage, and we grow about 50,000 plots right from F4 generation to F14 generations that are managed by this site. Talking about the major challenges that we focus on, fusarium is a major challenge for us. We have a very nice fusarium nursery that also does leaf rust, stem rust testing within the same nursery. And you can see a plot where the heads are discoloring because of the fusarium infection. These are the heads that show you a lot of fusarium infection on them. So this nursery is a really hot nursery for fusarium. We get good disease incidence. It's inoculated. And that's why we are able to screen out the bad germplasm from our breeding program. It takes about 1,500 kilograms of corn every year that we use for inoculation of this nursery. The nursery is about 30 acres, which is on 3-year rotation, and it's an irrigated nursery to maintain that inoculum of fusarium throughout the growing cycle. We grow about 50 to 60,000 1-meter rows, and this is not just from our program. We do it for the all AFC program as well as the university program. So Swift Current program, Lethbridge, University of Saskatchewan, University of Alberta, they all send their material to be tested in this nursery here. On top of these rows, we grow about 250,000 single F2 plants. They are space planted as individual plants, and they're all looked at individually. So it takes a significant amount of resource not to just create this nursery, but also assess it for diseases. We grow all the generations from F2 all the way up to F13s. So there is a lot of material at different stages of breeding, right up to the variety development, final variety development. And I talked about different participants. We also grow some international nurseries from CIMMYT who have an active FHB breeding program, and they have some unique germplasm with different sources of resistance. And we want to test that material in our nursery, and we do that so that we can find new sources of resistance that we can incorporate into our own programs. The next challenge is wheat midge. And again, our programs work actively on wheat midge resistance. So this is just the wheat midge pest. It should not be confused with another pest which is not harmful to the wheat crop. It's a little bit bigger in size to the wheat midge. Here is a comparison. And you can see that if the larvae are present on the spikes, they eat the developing grains, and it leads to yield loss. The major challenge is it's not just an yield loss. Whatever grains are remaining, they are also deformed, sometimes shriveled, and sometimes they have the larva present on the kernels. That makes poor grade for the grain, and the farmers don't get better prices for that grain. So that's why this pest is important. We have a single source of resistance, which is called SM1 gene, and there is no other source of resistance for this pest. So we manage this resistance because we have a lot of lines now with the SM1 resistance gene, and we manage it by putting a 10% refuge of susceptible lines in the resistant variety, and that's called a variety blend. So we have some lines from our program that are variety blend, and that is allowed to have something for the insect to eat within the field so that it doesn't develop resistance to that gene quickly. So that's an approach that has worked really well in our farming systems. There is active midge programs that are going on. There is a program called There Is No Plan B that is done by FP Genetics and ECAN. So we have had very good success, and internationally we are recognized for doing such an effort to maintain the resistance of a gene. So that's not just for financial benefit, but it also brings a good name to our country that we are able to manage this kind of farming practices. The other pest is Softline. At Brandon, we don't actively work on this pest because it's not a problem for us. And also, it's mandated to the Swift Current program to work on it. So Richard Cuthbert's program at Swift Current works very actively on this. This is a pest that leads to stem breakage. So there is lodging. Because of the lack of source to sink transportation, there is poor test weight, reduced yield, and there is no chemical control for this pest. So integrated pest management and genetic resistance because of the SST1 gene that is present provides resistance to this this pest, sorry. There are a number of lines out that have this kind of resistance to sawfly. Looking at the facilities that we have for breeding, other than the field and the nursery that we have a lot of growth cabinets that are used for clean increases of the seed. Let's say we make DH plants that needs to be increased. We use these facilities for a lot of increases. We do some disease testing. For example, loose must testing are done in these growth facilities. We have sufficient greenhouse space, and that's also used for increasing the seed. For example, if we have challenges going into the winter nursery, we can do some increases in the greenhouse. What you see here is an automatic watering system, and we are testing a lot of plants for bunt disease. So it's also used for testing of diseases. This is a representation picture of DH facility that we have in Morden. Modern facility generates about 2,000 lines for us every year, and that helps the breeding programs to get some of the varieties out faster because it gives us an advantage of about 4 years, up to 4 years, to develop a better variety. And usually the candidates for DH production are lines which are a cross between elite by elite, which means the line is really good but lacks one or few traits. We cross them and then we put it into DH pipeline, and that allows us to make better varieties a little bit faster than the traditional breeding. So it's a very good resource to have. Moving on to other breeding assistive technologies such as marker-assisted selection. So the markers are DNA fragments that are tagged to a certain trait. We don't actively have to phenotype something if we can run the marker in the lab. Phenotyping is time-consuming. You have to create the atmosphere to generate the right phenotype. We can bypass all of that by having the marker facility in our breeding program. So for example, sometimes very difficult to select for traits, those traits we can screen through markers. An example can be UG99 family of stem rust testing. Same thing here. It's also a quarantine disease, so we can actively test it out in the field. We do marker testing for it because we know if we have a certain marker in our germplasm, we have resistance to UG99 stem rust. Sometimes the breeder tries to gain few steps by making a complex cross. So you can bring in two parents and make a cross, which means you are only limited to the traits that are present in those two parents. But breeders do use a third parent sometimes, so they make a cross between parent 1 and parent 2 and take the F1 and cross it to a third parent. Now you bring in a few more traits that you can work with in your population. Those are called complex crosses, and these complex crosses can be run through the markers pipeline to weed out the traits that we don't want in that complex cross. And it allows us to reduce the population size going forward. So we are not looking at many, many thousand of twos in the field. We are looking at a fewer thousand. Also, it's used for pyramiding of genes. So if you're growing a plant in the field and you have resistance to certain diseases, but you don't know what the source of that resistance is or how many factors are causing that resistance in the plant. So markers allow to tease that out. Are there just one or two sources of resistance, or are there many sources of resistance in the plant? Because in the field, you just see the resistance. So it allows us to add more sources of resistance by knowing what is present in the plant and what can be added to the plant. And that's how pyramiding of the genes for complex traits works. And markers allow us to do that. We use winter nurseries for usually odd generation plants. So the F3s, the F5s, the F7 generation plants are shipped out to New Zealand right around September, October, and they are grown in New Zealand. We usually send about 16 to 20,000 rows to New Zealand. We go to New Zealand, we screen there for height, lodging, maturity, diseases such as leaf rust, stripe rust, fusarium sometimes. So it allows us a second cycle of selection and generation increase by having winter nurseries, and it makes that the breeding genetic gain a little bit faster. As I mentioned, the double haploid facility at Morden allows us to save a few years from the traditional breeding cycle that is done through selfing and selections. This is the serial genomics facility at Brandon, and this is designed to be high throughput and high efficiency lab, which means that we want to go through many plants per day, and we want to go through them really fast. That is the sole focus of this lab because we want to run markers really quickly through this lab, get that information so that we can eliminate the need of phenotyping in the field, which is time consuming and labor intensive. This machine here is an automatic DNA extraction machine, which means you have to grind your samples separately, but after that, this machine takes over and does the DNA extraction for you. This is the liquid handling system, which means you don't have to pipette as many tubes as you would do by traditional pipetting samples, either through a multi-channel or a single-channel pipette. This is an array of thermocyclers that is used to run the markers. And this image comes out of Swift Current, actually. I really like the way they have multiple PCRs arranged over multiple stacks, and they run a lot of markers through this. Once the markers are run, they are analyzed through this system, which is a spectrophotometer, and the result looks like this. So this is a test that is done for a leaf rest resistance gene, LR34, and you can see that there is a very nice separation of material. One is resistant, another one is not resistant, and you can easily see that, and you know what you have added to the plates here, then you can eliminate the ones that are not resistant, and that's a quick and easy way of screening the germplasm. So having the marker facility and the information on markers allows us to screen for diseases, and this is a great resource to have because it provides multiple levels of information for a breeder. So we know in a certain germplasm, it may, or a certain line may have all the genes that are shown here. But if we go to the field, we don't know what is actually causing that resistance. So knowing that, what is the potential or what is the genotype of the plant allows us to add more to it. So now we know that we have LR14, LR16, LR34. These are, LR means for leaf rust resistance. We know that we don't have LR46. Now we look for sources of LR46. We cross the lines with LR46 parents, and then we can screen those plants and make sure that now we have added the LR46 to it. So that's how the marker helps. You can also see that there is a series of different types of gene written together with slashes. So this LR34 YR is stripe rust resistance, SR is stem rust resistance, and PM is powdery mildew resistance. They are written with slashes because there is one marker that shows the presence of all of them. So we know that we are having leaf rust, stripe rust, stem rust, and powdery mildew resistance all by testing one marker. That's another advantage of having a marker system in place. We have markers for stem rust, leaf rust, stripe rust, fusarium head blight, loose spot, tan spot, and we actively use that in our breeding program. These are some of the important varieties that are currently in the marketplace or are coming to the marketplace very soon. AAC Warman comes out of our program. I did not mention the traits because if it is a registered variety, it has passed those minimum requirement of becoming a variety. So it has good yield. It has decent resistance to fusarium. It has all the quality traits that you need. It has resistance to all the rust that you need. But I wanted to show you why that knowing the genetic makeup of the plant is critical. Now we know what all types of resistances and some of the ones that we cannot take, for example, the waxiness or the vernalization pattern of this line. So we know some of those traits now by having the marker information of this line. We already know it has all the good traits, it will be a great variety, but now we also know what it can be crossed to so that it can provide the resistance that it has to fusarium because of the 4B to another line that we are trying to develop. So this is some of the information that we can capture out of the new lines. And this is another aspect of our program here at Brandon, which we actively do, is try to genotype the newer lines for new sources of resistance that it may have. AAC Brandon is one of the lines that is commanding a lot of acreage in Manitoba. It's at about 65% in Manitoba and about 35% all across the prairies. It's a great line, and now it has in here. One thing I would say that it was rated poor for sprouting, so there should be some caution in growing this under different conditions. Leroy is another one that is coming out of our program, and we know that it has SM1 resistance. It is a line with Alliance seed, and hopefully it will be available to farmers next year. AAC Wheatland comes out of Swift Current program from Richard Cuthbert's program, and it's also a great line for abiotic stress regions. Here's another great example of a line that came out of our program and should be available through FP Genetics in the next 2 years, is that it was rated really well for fusarium resistance. But when we looked at what was the genetic makeup of this line, there is no fusarium resistance gene in it. So what is causing that resistance? We have tested it over and over multiple years, and it was always very good resistance, but we don't know what that resistance is. Now this becomes a research study for us to figure out what the new source of that resistance is, and then incorporate it into more plants and more lines. So we do some of that research at Brandon as well. Moving on to the innovation part of our program, the biggest resource for a breeder is the germplasm. So we keep our eyes peeled all the time if somebody is talking about germplasm. And we don't restrict ourselves to just Canadian germplasm. We look at the North American germplasm as a whole, especially from the northern United States. We look at CIMMYT for a lot of the resistant genes that they find, and they are way ahead in terms of disease resistance from us. So there is a lot of germplasm we can use from them to bring in the resistance in our germplasm. We look at Europe. So Pasteur is one of the lines that was developed in Europe and is now grown here. We look at Southeast Asia to bring in blast resistance. We look at the disease hotspot zones. For example, we look at Kenya for UG99 resistance. We look at Bangladesh for wheat blast resistance. And what resistance lines are grown there, we bring them over here to bring in that resistance from those zones. We also have access to quarantine nursery. For example, there is one quarantine nursery level 4 at Morden where we test the UG99 at seedling stage within Canada. You cannot do that testing anywhere in Canada out in the open. So having that kind of facility helps us immensely to be ready when the disease is here. We also bring in lines from abiotic stress zones, for example, Israel. I was talking to a breeder from Israel, and he said, you don't have heat stress. You have minor inconvenience. We have heat stress here at Israel. So we bring in germplasm that have really high stress resistance from the zones that are experiencing high heat stress. The new area of science that we are looking at is phenomics. And within phenomics, the key is to phenotype as many plants as we can in a high throughput way. Just like markers allows us to genotype plants, phenomics allows us to phenotype plants in a high throughput manner. So there is a lot of different types of technology, both ground-based and aerial vehicles, like this drone here, that does a lot of imaging of the plants. And it allows us to not just see the plant in the regular colors, the RGB colors, but it also uses hyperspectral imaging that this gives us an insight into the physiology of the plant. So we're not just looking at the color of the plant, but also looking at what is the physiology of the plant indirectly. And it has a lot of different uses going forward when we combine this technology with the genomic technologies that we have. And it has a lot of potential to make the genetic gains faster for us. Moving on to genomic selection, that is the new area of science that has a lot of promise for breeding. And just to compare what we do in genomic selection compared to the traditional breeding, in traditional breeding we make regular crosses, as I showed in one of my slides. We developed self lines which are called inbred lines. We put them into small-scale trials. We collect a lot of data. We push them into larger trials. And in larger trials, we mean multi-location registration trials. From that large-scale trial, we find varieties that are really promising, and we release it as varieties, and we also use them for making new crosses, because they become parents for us. In case of genomic selection, we add a few more steps. And this is how genomic selection pipeline looks like. So yes, we make the same crosses there. If we want to accelerate the cycle, we push it through the double haploid pathway because it allows us to gain a few years. Then we do the genotyping, which means we know what markers, what genes it has within it. This genotyping information is fed into a model, a kind of regression analysis that is used to predict, and I will explain. From genotyping, we move on to the small-scale trials, then the large-scale trials, and the information from the small-scale trials, now this is the phenotypic information, from these two trials are fed into the model. Now, the model has both the phenotypic information and the genotypic information. And that's sometimes called a training set. So you do this a few years in your breeding program where you try to capture the information of the entire breeding program, both genotypically and phenotypically. So the potential and what it shows in the field. This model now becomes a predictive model so that in the future generations within your breeding program, you don't have to do extensive phenotyping and you can just predict what kind of yield, what kind of disease resistance, or what kind of quality one would get as long as the lines are similar to what was used to generate the data. So if I do this for a few years within my germplasm, within my breeding program, I can safely say that I will have good success using the predictive model without doing any phenotyping, what my lines would look like. And that's the promise of genomic selection. It hasn't been implemented in major breeding programs yet, but all the breeders are moving into it, slowly trying to figure it out, what is the best and cost-effective way and the easiest way to implement it in their breeding programs. There may be other sets of data like the physiological data, stress resistance data, that can be added to the model, and the more data that is fed into the model, it becomes a lot more better at predicting the values of the variety. And still doing all of that, you still get to the varieties, but hopefully 4 years faster compared to the traditional breeding over here. So, to summarize what breeding was, or what breeding is, within our breeding programs in Canada, we have made some respectable genetic gains. Within the CWRS, we are at about 0.67% yield every year. This is despite all the restrictions of quality that we have. The export potential should not be affected. The pre-harvest sprouting should not get affected. We have to keep pushing the yield while maintaining the quality and the disease resistance of the crop. Fighting all the new races of diseases, we have been able to make those genetic gains. And I think that's pretty good for what our previous breeders have done, and hopefully we'll be able to continue that in the future. Can we go back? We are looking at a lot of the upstream and delivery science by doing physiological studies, phenomics, genomics, and all of that is getting slowly incorporated into our breeding program, and we hope to make good gains, better gains, faster. There is a lot of technology, such as genetically modified crops or CRISPR-Cas technology that is available to breeders. There will be faster gains made by them, but unfortunately, we don't have the social acceptance for the lines that are developed through those pipelines. And hence, the breeders are kind of leery in using those technologies as of today. But in certain parts of the world, this is getting a lot more acceptance, and we hope that this becomes something that is a useful technique for us. I also appreciate the need-based innovation and the funding that the farmers provide to us. They need better varieties. There is a lot of funding to do that kind of research, and I really appreciate that. So thank you very much. So this is our team at Brandon Research and Development Center. This is the biggest resource for any breeding program the very highly skilled and motivated, dedicated staff that work really hard in the field and out of the field to make the varieties. It's a long process. It takes about 12 years in a traditional breeding cycle to see the fruit of the labor that is put forward. So I really appreciate having a really good group of people to work with. Dr. Jennifer Mitchell-Fetch is an oat breeder. Dr. Kirby Nelson is the new scientist who has joined us in the cereal science, and he'll be doing breeding and phenomics. We have a lot of senior technicians here that know the program, know the breeding germplasm, and are extremely helpful in keeping us out of trouble. Let's just say that. And at last, I would like to thank all the funding agencies, especially the Canadian Wheat Research Coalition, all the provincial funding partners, for all the support that you provide us to do the work. Thank you.
Is there any questions?
I must have done a really terrible job or a really good job. Thank you very much.
On behalf of Manitoba Wheat and Barley Growers Association, I'd like to present a little token of our appreciation for putting on an excellent presentation. Thank you, Dr. Kumar.
Thank you. Thank you very much.